The overall goal of this procedure is to isolate viable cardiomyocytes from diseased human ventricular specimens and characterize their functional alterations. This is accomplished by first rapidly collecting and mincing fresh ventricular samples in ice-cold cardioplegic solution in order to avoid excessive tissue degradation and cell damage. The second step is to perform a stepwise digestion of the myocardial chunks using a custom-made digestion device and enzymatic solutions in 6 cycles. The cell-containing buffer is collected in a tube at each cycle and diluted with a cell-preserving solution. The final step is to resuspend the cells contained in the 6 tubes in a lower volume of standard physiological solution with normal calcium concentrations in order to perform the functional characterization. Ultimately, patch clamp recording of action potentials and simultaneous assessment of intercellular calcium using fluorescent dyes is used to show the alterations of action potential duration and intracellular calcium cycling in cardiomyocytes from patients with hypertrophic cardiomyopathy.
The main advantage of this technique over existing method, like the standard chunk digestion method, is that, in association with the enzyme solution, we use a custom-made digestion device which allows a gentle dissociation of viable myocytes thanks to its silicon scripts.
This method can help answer key questions in the field of cardiology, such as how the well-known molecular and structural remodeling associated with cardiac diseases reflects into alteration of single cardiomyocytes that can be analyzed by our method.